Good morning, children. Welcome to Class 10 Biology, lesson number 23, Ascent of Sap. In this chapter, we will be discussing the structure of the root and what makes it immensely useful for absorbing water. Also, the different methods by which water is absorbed from the soil and transported to the adjoining cells. But first, let us study the function of the roots. Roots play a very important role for the plants. They help in fixing the plant to the soil. They also absorb water and minerals from the soil and transport it to the upper parts of the plant through specialized tissue called the xylem tissue. Next, let us study the structure of the root tip, which is very important for absorption of water. The root tip, the purple colored part of the root tip, is the root cap. The root cap is responsible for protection of the growing tip from any type of mechanical injury as the root tip moves deep into the soil. The first zone, which lies below the root cap, is the growing zone, a zone of cell multiplication, or the meristematic zone. Here, the cells keep on dividing continuously. The next zone is the zone of elongation. Here, the cells elongate and attain their full size. The third zone is the zone of maturation. Here, the cells mature according to where they are placed. That is, the cells of the epidermal layer form the root hair. The cells which are placed where the xylem vessel should be form the xylem vessels. So, this way, the outermost portion form the root hair which is responsible for absorption of water. What is the role of water in plants, or why do plants need water? Plants need water for transportation. Water is a suitable medium which causes transportation of substances in solution from the roots to the leaves and the food prepared by the leaves to every cell which is non photosynthetic, that means which cannot make its own food. Next is photosynthesis. Water is the raw material for synthesis of glucose. We have already studied it in the previous chapter. Through photosynthesis. Mechanical function. We all know that plants do not have a skeletal system. Hence, water provides turgidity to the plant tissues. Transpiration. A large amount of water is lost as vapors during transpiration, but this helps in creating a suctional pull by which water reaches to the top of the plant, right, even if it is some 50 to 60 feet plant. Stomatal movement, opening and closing of stomata, is also one of the functions of water. Maintaining plant temperature. Water has a high specific heat. Hence, the plants can absorb a large amount of heat without any major rise in temperature. Now, what is the role of minerals in plants? Let us study some of them given below. Magnesium. It is essential for synthesis of chlorophyll. Without magnesium, chlorophyll cannot be manufactured. Zinc. It is required for leaf formation. Calcium is essential for maintaining semi-permeability of the cell membrane. Potassium controls the opening and closing of stomata. We have discussed this in the potassium ion concentration theory in the previous chapter. Manganese, it is essential for photolysis of water, that is breaking down of water into H and OH ions. Nitrogen is necessary for synthesis of proteins. Let us now study the characteristics of roots for absorbing water. Roots have an extensive root system. It is so believed that a 4-month rye plant has a root system of 620 kilometers and it increases every day by 5 kilometers. This enormous root system has an extensive or enormous surface area for absorption of water. Next is high concentrated solution. The root hair have a high concentrated cell sap. This helps in drawing water from the outside into the root hair by a process called osmosis. The thin walls of the root hair are most efficient region for absorption of water. This region is the region of root hair. In this region, there are numerous root hair. Root hair are specialized for absorption of water as they have a thin cell wall. You can see in the figure the root hair in different stages of its development, and even you can see that the outer covering, that is the cell wall and the cell membrane, is very thin. The cell membrane is semi-permeable. But before we proceed, let us see the different types of membranes that are present. Permeable membranes, membranes which show diffusion of both solute and solvent molecules through them, are known as permeable membranes. Cell wall is a permeable membrane. Impermeable membranes, membranes which do not allow diffusion of either solute or solvent through them, are known as impermeable membranes. Semi-permeable membranes, these membranes allow diffusion of solvent molecules through them, but not solute molecules. Then differentially permeable or selectively permeable membranes, membranes which allow useful substances to enter inside and harmful substances to leave them, are known as selectively permeable membrane. Cell membrane is selectively permeable. Modes of absorption and conduction of water and minerals. Absorption of water and minerals from the soil and their conduction involves a number of processes: osmosis, imbibition, diffusion, turgidity and flaccidity, plasmolysis and deplasmolysis, passive transportation and active transportation. We will discuss each one of them one by one. First, we will discuss osmosis. What is osmosis? It is the movement of water molecules from a region of higher concentration to a region of their lower concentration when they are separated by a semi-permeable membrane. You can see that line that you see, the black line, that is the semi-permeable membrane. It allows the water molecules, only water molecules or solvent molecules, here it is water, to pass through it, not the solute molecules. Okay. Osmosis is of two types. Endosmosis, it is the inflow of water into the cell or inflow of water into a system, that's called endosmosis. And exosmosis, it is the outflow of water from the cell to the outer surroundings or from a system to its surroundings. Let us study this with the help, understand this with the help of an experiment. Set up a thistle funnel as shown in the figure. Inside the thistle funnel, in the first arrangement, that is setup 1, place sugar solution and cover the end of the thistle funnel with the help of cellophane paper. Now, place this thistle funnel in a beaker containing pure water. Suppose the concentrated solution in the thistle funnel is say 20%. What does this mean? 20% is solute and 80% is solvent. That means there are 80 molecules of water inside the thistle funnel and outside is pure water, that is 100% water. In the second setup, what we do is just the reverse. We are going to take sugar solution, say 30% concentrated sugar solution in the beaker, and place pure water in the thistle funnel. Again, the mouth of the thistle funnel is covered with the cellophane paper. Now leave this arrangement for 2-3 hours. What do you observe? In setup 1, the level of solution rises. In setup 2, the level of the solution falls. Now what is actually happening in both these arrangements? In setup 1, the concentration of water is more in the beaker because there is 100% water there, and there is 80% water in the thistle funnel. So water enters from its higher concentration, that is from the beaker, into the thistle funnel, and hence the water rises in the thistle funnel. In setup 2, the concentration of water is less in the beaker and more in the thistle funnel, because the thistle funnel has pure water, and in the beaker there is a concentrated solution, say 20% concentrated solution of sugar. Hence, the water moves from the thistle funnel to the beaker. This is known as exosmosis. Okay, in the previous one it went inside, so it was known as endosmosis. Here it is moving out, hence it's known as exosmosis. Now let us study this with the help of a potato osmoscope. Take a potato and peel the skin. Then scoop off the inner portion as shown in the figure. Place a concentrated solution of sugar, say 25% sucrose solution, inside the cavity of the potato and place the potato in a petri dish containing pure water. Leave this arrangement for some time. What will you observe? The level of solution in the cavity rises. We can see it in the figure. Inference: the level of the solution in the cavity of the potato rises because of osmosis. The water molecules diffuse from the petri dish through the semi-permeable membranes of the cells of the potato and pass into the cavity, which has low concentration of water. That is, it has 75% water, and outside there is 100%. Hence, the water moves from the outer petri dish into the potato osmoscope. Now, with this in our mind, let us see the next experiment. According to rule, the water should enter into the thistle funnel or into the potato as long as the concentration on both sides is not equal. But when you actually go about with the experiment, this does not happen. Now, let us discuss why it does not happen and what is this term called. Osmotic pressure, the minimum amount of pressure that must be applied to prevent the passage of pure solvent into a solution through a semi-permeable membrane, is known as osmotic pressure. In this experiment, we are going to demonstrate this atmospheric pressure. In the first setup, we have the thistle funnel, which has 20% sucrose solution in it, and it is placed in a beaker containing pure water. And you will see that water starts entering into this thistle funnel, and the level of water rises. After a period of time, even though the concentration of water on both sides of the thistle funnel of the cellophane paper is not the same, the entry of water stops. Now, in the second setup, we take a similar thistle funnel with the same material, but now we place a piston in this. And we will find that as soon as water starts entering into the system, we start putting weights on the upper portion of the thistle funnel. Care should be taken that the piston lies just in contact with the sucrose solution. Now, as you keep on adding weights on it, you will find that now no further entry of water is taking place into this. Inference, or what do we understand by this? This is because the solute molecules which possess motion and kinetic energy distribute themselves in the entire available space by the process of diffusion. They then exert a pressure on the solution which is known as the osmotic pressure. In short, we can say that the osmotic pressure is equal to the weight or the pressure required to nullify osmosis. With this, we come to an end of this class. We will discuss the other different methods by which water is transported from one cell or goes from one cell to another in our next class. Thank you, children. Take care of yourselves, and God bless you all.